later. Another very important characteristic or function of a DNA molecule is that it is a molecule responsible for the formation of proteins in our bodies. We know that for a healthy life, our bodies need proteins. Yes, we get proteins from the food that we eat, from meat, from milk, from beans, you name them. But our bodies are capable of forming its own proteins. And this process is facilitated by a DNA molecule. The DNA molecule is the one that will determine which protein needs to be formed. It is the one that determines the type of a protein as well as when and how. To do this process, DNA molecule needs an assistance from RNA. They work together to form proteins in our bodies. Okay, let's look at this diagram here. What happens during the formation of proteins? One other thing that I did not mention earlier on is that the pairing of the nitrogenous bases, meaning the purines and the pyrimidine, is done in triplets. We call that a triplet of bases, meaning they are being grouped in threes. It can be guanine, cytosine, thymine, cytosine, cytosine, guanine to make the three. And we call that triplet of bases. As you go along, there's another grouping. They're grouped in threes. We call that triplet of bases. It's very much important to know this, because when we are going to discuss protein synthesis, you will see the importance of this. Now, what happens during protein synthesis? First of all, proteins are formed by amino acids. We have got 20 different amino acids. And these amino acids are used to form different proteins in our bodies. Another thing to note is that this process of protein synthesis takes place in the ribosomes. Let's go back to the structure of the cell. Go back to your grade 10 topic on the structure of a cell. Remember, we have the nucleus, we have the cytoplasm. In the cytoplasm, that's where we find other cell organelles. We have the ER, endoplasmic reticulum. And in the ER, some of them have some little dots called ribosomes. It is in the ribosomes where proteins are formed. So proteins are formed in the ribosomes. Now remember, I said the DNA, which is located inside the nucleus, it is the one that has got the coded message about a particular protein to be formed. Now one will wonder, then how does the nucleus, how does the DNA take this message to the ribosomes and the process of protein synthesis starts or proteins are formed. This is something that you are going to learn here, and you will understand why RNA is needed during this process. Okay, look at this diagram here. This is the same DNA that I was talking about. It has got 2 strands. It's a double helix, unlike RNA. Now, what happens during protein synthesis? The DNA the 2 strands of DNA will separate. Remember, I said it will unzip, meaning the hydrogen bonds will break one by one. And at the ultimate end, we will have 2 separate strands of DNA molecule. And one strand of a DNA will act as a template. Look at this diagram here. This is strand number 1. This is strand number 2. The 2 strands are now separated because the hydrogen bond is broken. So one strand of a DNA molecule will form or will serve as a template forward for the formation of messenger RNA. So messenger RNA is formed according to the template maintained by a DNA molecule. Remember, I said DNA molecule has the message, the coded message about a particular protein. And I've also indicated that these purines and pyrimidines are sort of grouped together. They are triplet of bases. And the way they are arranged will determine a particular message. You'll understand as we go further, looking at the schematic representation of the process. Now, here is DNA molecule. The strands are now separated. Here is the messenger RNA developing. And how? Just like the DNA molecule, which has got nucleotides in the nucleoplasm, same applies to RNA. In the nucleoplasm, there are loose nucleotides for RNA molecule. So they are going to arrange themselves according to the template maintained by DNA. How does this happen? Let's go to another slide. But before then, you must know that this strand, it's a DNA strand. Somewhere, somehow, there will be T, which stands for thymine. And there will be A, which stands for adenine. Now, when the RNA nucleotide arranges itself along this template maintained by DNA, instead of thymine, there will be uracil. And adenine from the DNA molecule, instead of adenine and thymine, it will be adenine and uracil from the side of a nucleotide for RNA. let's look at this to make things easier for you. Right. One strand of mRNA has been formed. Let me go back. I'm coming back here. Here is a stand for RNA. It is now formed. And as this strand is formed in accordance with the strand of DNA, we say there is copying of message. The message is being coded. And this process is called transcription. Why do we say transcription? We're actually saying the DNA has got the message, it's got the coded message about the protein to be formed, and then a messenger RNA, when it is formed, it sort of transcribes, it sort of copies the message. After copying the message, after messenger RNA is formed, the messenger RNA will leave the nucleus. Why does it leave the nucleus? Because it carries within it the message about a protein to be formed. And I said protein synthesis takes place in the ribosomes outside the nucleus. So this messenger here, which is messenger RNA, has copied the message. It is now leaving the nucleus to the ribosomes where proteins are being formed. Now here is the ribosome. This big orange structure is the ribosome. And the messenger RNA now, it is going to attach itself on the ribosome. Remember, it carries the message about a protein to be formed. Where is this message coming from? It is coming from the DNA molecule. How was this message copied? That is when the two strands of a DNA molecule separated. Messenger RNA is formed according to one strand of a DNA molecule. By so doing, the message is being copied, and we refer to this process transcription. Then when this messenger RNA has attached itself on the ribosomes, carrying the message about a particular protein to be formed, the transfer RNA, remember I said we have three types of RNAs, the messenger, the transfer, and ribosomal. Now the transfer RNA is the one that will select amino acids from the cytoplasm. I said we've got 20 different types of amino acids. Now the message carried by the messenger RNA will dictate which amino acids are needed for that particular protein. So the transfer RNA will collect amino acids from the cytoplasm and transfer them to the ribosomes where proteins are being formed. Now one will ask this question. Remember, it is the DNA that will determine which types of protein, which types of amino acids are needed for that protein, and how should these amino acids be arranged, and how many amino acids are needed. Let me repeat again. The copied message from DNA has got the following. It indicates, number one, which amino acids are needed for the protein. Number 2, how many of such amino acids are to be used for such a protein. And number 3, how are those amino acids to be arranged? The arrangement of such amino acids to form a protein. Now, when the transfer RNA collects amino acids from the cytoplasm to the ribosomes, there will be no mistake. The amino acids that are being selected will be placed or arranged in the sequence that was determined by DNA and the messenger RNA. Why? Because of the triplet of bases that I talked about. The triplet of bases from the messenger RNA is called the codon. The same triplet of bases appear in the transfer RNA, and it is called the anticodon. I'm repeating, the triplet of bases that I talked about earlier on are found in the messenger RNA, and we call that the codon. Same triplet of bases are found in transfer RNA. It is called anticodon. So to make sure that this is the correct amino acid, it must be put here. That is determined by the codon and anticodon. The transfer RNA will fit in where its anticodon matches the codon of the messenger RNA. You'll see that when we look at the schematic representation of the whole process. Now, the transfer RNA will pick up as many amino acids as required according to the message, and they'll be arranged in the required manner. And when 2 amino acids are put together, we call that a dipeptide. This is a process that ends up with the release of water, process called condensation. And when another amino acid is added, it's a tripeptide. If more and more amino acids are added, we talk about a polypeptide. Now, plus minus 50 amino acids are needed to form a protein. So this will be the role of a transfer RNA to pick up as many amino acids required to form a particular protein from the cytoplasm. Look at the shape of the transfer RNA. The structure on your right is the transfer RNA. This yellow parts indicate the anticodon. It's a triplet of bases. Remember when I talk about triplet of bases, it will mean maybe this is CGU. There are always 3, but in this case here, it is CCG. So when an amino acid is being picked up by this transfer RNA and taken to the ribosomes, for it to fit in the right place, the codon of the that the anticodon of the transfer RNA must match with the codon of transfer RNA. Call this the 3-base sequence. Please don't forget the structure, because in the exam papers, this is the structure that they normally use to indicate transfer RNA. Fine. This is just the repetition of what I've said. Here is the ribosome, the messenger RNA attaching itself here with the message about which protein is to be formed, which amino acids, how must they be arranged, and how many amino acids are needed. So this is the transfer RNA with the anticodon. This triplet of bases from the messenger RNA is called the codon. This one, the below one, is called the anticodon. So finally, we'll have amino acids that are being selected and arranged in the required manner for the formation of a protein. Now, this process whereby amino acids are being picked up by the transfer RNA, taken to the ribosomes, where they are being arranged to form a polypeptide, which can be called a protein when it has got 50 amino acids. It's called translation. I'm going back to what I said earlier on. I said when messenger RNA is formed inside the nucleus, when the DNA strands are separated, messenger RNA is formed, and then loose nucleotide of messenger RNA arrange themselves in accordance with one DNA strand, which serves as a template. The messenger RNA is formed. The message about a protein is being copied, and that process is called transcription. The message has been transcribed, has been copied. It is now taken out of the nucleus to the ribosomes. Now, here is the process whereby that message is being translated into bringing in types of amino acids that are needed for this protein, how they are to be arranged, and how many are needed. So this is translation. So these two terms are very important. You are sometimes requested to discuss what transcription and translation mean. This is what we have said already. It's transcription, when the message is being copied, when amino acids are being collected and transported to the ribosomes according to the message, we refer to that as transcription. So transcription is a process that is enzyme controlled. The whole process whereby the DNA molecule unwinds, two strands separate, messenger RNA is formed, is controlled by enzymes. The helicase enzyme is the one that facilitates the breaking of the weak hydrogen bond. And the polymerase enzyme is the one that facilitates the addition of loose nucleotides to the DNA strand to form the messenger RNA. Okay, let's look at this diagrammatic representation of protein synthesis. This is the nucleus. In the nucleus, there is a DNA molecule. Remember, the DNA is found in the chromosomes, and a section of the DNA is called a gene. When a protein is to be formed, the message comes from the DNA. It will unwind, the two strands will be separated. One strand will be used as a template to form messenger RNA. This process is called transcription because the message now is being copied. The coded message from DNA is being transcribed through the formation of the messenger RNA. After this message has been copied, messenger RNA leaves the nucleus carrying the coded message from DNA to the ribosomes. And in the ribosomes, remember I said this is the cell organelle where protein synthesis takes place. So the transfer RNA, remember this shape, will transfer amino acids, collects and transfers amino acids from the cytoplasm to the ribosomes where proteins are being formed. So the message now is being translated. Which message? The message that comes from DNA that was carried by the messenger called messenger RNA. The tRNA brings amino acids to the ribosomes. Where do we find amino acids? In the cytoplasm. They're just free. And transfer RNA will pick up the amino acids and transfer them to the ribosomes. So a chain is going to be formed. Remember what I said? As many amino acids are being added, a polypeptide will be formed. The first two will form a dipeptide. When we add another one, a tripeptide. When we keep on adding many amino acids, a polypeptide is going to be formed. Okay, before this, I just want to go back here a little bit. And I want to hammer on this fact. To say, look at the messenger RNA here. It has got triplet of bases. The triplet of bases, which is called the codon, will match the triplet of bases of transfer RNA, which is called anticodon. Meaning, for an example, learners, if this triplet here is CCG from the messenger RNA. If this is CCG, remember, C is for cytosine and G for guanine. So the transfer RNA that will fit in this triplet of bases, which is called codon, must have G, G, and C. Remember the lock and key order. The shape needs to match here. The shape of the purine needs to match that of a pyrimidine. And do not forget that, for an example, the triplet of bases for the RNA, it is maybe A, U, G. U, G. You must always remember that A, which is adenine, will match with uracil. So in an exam paper, they can give you these triplet of bases just to want to see as to whether you know that in RNA there is no thymine. They'll give you a triplet of bases having thymine and would want you to select which one will match the messenger RNA. Be careful, there's no thymine. This is RNA, there's no thymine, there is uracil. Okay, what is the importance of proteins? They serve as building blocks of cells. They control chemical reactions and transport materials to and from cells. You remember what I said earlier on to say we need proteins for our bodies to be healthy. So the proteins that are being formed by our bodies are not enough. Hence, we need to eat as much protein-containing food as possible to keep us healthy. Thank you so much, dear students. We'll meet again.